Welcome to the Perth Eye Foundation 2021 lecture. My name is Ross Littlewood. I'm an ophthalmologist in Perth and I have a special interest in clinical colour vision testing. The Perth Eye Foundation is an independent charity that distributes funds for ophthalmic philanthropy, education and research, both nationally and internationally. Imagine I said to you, the grass is green. You might interpret me as meaning that there is a property called greenness inside the grass, or perhaps on its surface. That view is called naive realism. In the 19th century, that view was challenged by the discoveries of Thomas Young, James Clerk Maxwell, and Hermann von Helmholtz, who all contributed to the trichromatic theory of colour perception. However, realism is very resistant to scientific advancement, and a different view evolved called colour physicalism, which holds that there is indeed a property in grass which determines our perceptions, and that there is an uninterrupted chain of processes from pixel to perception. However, in individuals who perceive any one given stimulus, it's quite possible that one of these three channels that transmit the physiological perception could be anomalous, so that it's possible we could have different versions of the greenness of grass. Nevertheless, it is the end result of a bottom-up process. Of these three thinkers, Hermann von Helmholtz was the most skeptical about the capacity of the human eye to fully represent over 1 million different colour perceptions and to account for phenomena such as colour constancy or the various anomalies that psychologists have illustrated, including the dress paradox seen on this slide. The alternative view is colour idealism, according to which colour consciousness is a separate process to the bottom-up series of processes that lead to colour contrast in the outside world. So consciousness is a top-down mental construct, and idealism is a term of craft in philosophy that refers to such processes. There is quite a lot at stake for science if colour physicalism is not true, because scientific conversations that refer to nothing but facts outside of the observer are only self-referential and at worst not even truth-apt. Consider a single pixel from image 2 in this slide. If we take that pixel and draw its spectrophotometric curve, we have done the best job we can think of at displaying its spectral content. However, even that effort doesn't uniquely define the stimulus, because dozens, if not hundreds, of different spectrophotometric curves can produce the same identical colour perception and also the same physiological response to the input. Equally, if we take any single pixel from the picture and show it under a variety of different viewing conditions, we can get a range of different perceptions. Those variations in viewing condition could include duration, brightness, pre-adaptation, background contrast, and so on. Now consider the photoreceptor array illustrated in picture 4 in the bottom right. You'll see that there are relatively few blue cones and none in the central retina. Below that, we have a schematic of a cone in red, and on the left, an analog input signal, which is optical, and on the right, a digital output signal, which is electrical, representing one or two charge states by each photoreceptor. When any analog to digital transducer undersamples the input frequency, an aliasing artifact occurs, as shown in picture labeled 5. The critical sampling rate has to be at least twice the input frequency. Although the phototransduction of 11 cis-retinol takes only around 200 femtoseconds, which is blindingly fast, if you'll pardon that expression, the reconstitution of all the plasma membranes in the outer receptor of the cone is quite time dependent, and after a complete bleach takes several minutes. So aliasing probably does occur even in the red and green cones, but certainly occurs in the blue cones and rods, which respond much slower. However, that would not cause a distortion in spectral perception, but rather a decrease in sampling. A different type of sampling error occurs when we consider the ability to represent a spatially distributed optical image by a finite array of pixels, such as the cone array. If we undersample the image, then we get a phenomenon called Moir patterns. There are several tests to demonstrate that the human retina does produce Moir patterns. One is to use laser interferometry, in which lasers of different wavelengths are used as inputs, and the grids are rotated so that observers have to select the axis of the lines in the interference grid. When long wavelength lasers are used, observers can reliably detect the correct axis when the frequencies go above 50 cycles per degree, sometimes up towards 60 cycles per degree. However, when short wavelength lasers are used, such as blue, the normal observer seldom achieves correct identification of the axis at frequencies of 15 cycles per degree, even if they can tell that it is a blue signal. That is because the spatial resolution is simply too low in the blue system, as we see when we look back at image number 4. The absence of blue cones in the center and their relative paucity around there, and also the fact that they respond slower than the red and green, is thought to be the explanation for these phenomena. This animated GIF demonstrates what Moir patterns look like. It's a form of aliasing, and yet in everyday life we tend not to see these Moir patterns. One reason is that our fixation behavior involves a series of very rapid saccades from one point on the retina to another, rather than persistent fixation from one finite pixel. There's also slow drifting movements, and the combination of these phenomena means that what would otherwise be distributed patterns is transformed into a series of interrupted transients. Now we'll continue our bottom-up journey following this pixel to perception by examining the contents of the optic nerve. 80% of the optic nerve fibers are parvocellular ganglion cell fibers, and they contain all the information about luminance contrast other than the high temporal frequency signal that is in the magnocellular fibers, and also all of the information about red-green chromatic contrast. Those parvocellular fibers terminate in the parvocellular parts of the lateral geniculate nucleus, and from there relay to layer 4 of V1, or visual cortex. In V1, some of the fibers congregate into what are called blobs, which stain with cytochrome oxidase. The coniocellular output numbers only 8% of the ganglion cell fibers in the optic nerve. They are thinner, more fragile and susceptible to disease, and also stain differently to histochemical stains than the parvocellular fibers. They are also a diverse population. Some accompany the intrinsically photosensitive retinal ganglion cell fibers to the tegmentum, where they supply the input for non-conscious visual responses. These include circadian rhythm, phototropism, visual attention, pupil responses, and release of melatonin. There's also a subset that travel directly to the middle temporal area called V5 without relaying in V1, and the fact that that output is preserved under conditions of cortical blindness is thought to be the reason that some people who are cortical blind can identify which side of their body movement is occurring on, a phenomenon known as blind vision. But probably the most important subset of the coniocellular output for us is the group that accompanies the parvocellular fibers into the lateral geniculate nucleus, where they remain functionally and anatomically separate. They then relay to a different layer of V1, where once again there is no unity of perception. And a total of 50% of the fibers sampled by electrodes in primate brains are found to be chromatically responsive and 80% luminance responsive, but the vast majority are red-green contrast responsive. An interesting thing occurs in the feedback from V1 to the lateral geniculate nucleus. The parvocellular signal is aggregated into a corticofugal pathway that leaves V1 in layer 6 and has an inhibitory function on the bottom-up input into the lateral geniculate nucleus, but it is entirely pathway selective. That is to say, the induced response inhibits the bottom-up parvocellular response without affecting the coniocellular response. Because it is temporally delayed, it's able to neutralize those transients that we mentioned earlier when talking about Moyer patterns. And because it is spatially averaged, it's also able to filter out diffuse background glare, such as is seen in early cataract or phenomena where there's a problem with a patient's intraocular lens, where they surprisingly don't notice much of the scattered light on their retina. One advantage of filtering out constant scatter is that upstream processing would have required a lot of mental resources, but that's now filtered out of the system, allowing the observer to simply concentrate on what is important. Next to V1 there is V2, and in that part of the brain the cytochrome oxidase standing cells are arranged in stripes, and they are also colour responsive. On the inferior aspect of the occipital lobe there is V4, and that seems to have different functions in primates and humans. In humans, functional MRI testing shows that there are chromatically labelled inputs, and also pathological studies on humans show that ablation of that area, for example in strokes or tumours, causes a phenomenon called cortical colour blindness. However, surgical lesions in that area in primates do not cause cortical colour blindness, which indicates a difficulty for colour research because it's one of many examples of where physiological studies in animals do not correlate with psychophysical responses in human observers. Beyond V4, the signal bifurcates into the dorsal and ventral streams, as illustrated in image number 8 at the bottom of the slide. The dorsal stream to the parietal lobe allows us to represent the world in an egocentric map, as opposed to a Google Earth map. Our egocentric map populates our immediate environment with nested volumes, any of which is a potential object of regard. The ventral stream radiates towards the temporal lobe, and in the temporal lobe, the object of regard is interrogated to answer the question, what is it, in a categorical sense? One of those categories being, of course, colour. Electrodes placed in primate brains in this area do show responses to what is called unique hues, that's red, green, blue and yellow, but not to all the other hues that a person may perceive. And those unique hues are found to differ a little bit between humans and primates. The input to the temporal lobe allows an observer to aggregate different sets of, or different components that form a whole into one gestalt perception. For example, when a solid object that's distributed widely across the visual field is camouflaged by a foreground. This could, of course, occur with a line in the undergrowth. This slide shows two circles at the bottom that have very finely striped stimuli, one green and one yellow, and the animated GIF will interlace them. So the observer now sees an intermediate perception. However, there is no pixel with that colour on the page. An analogous phenomenon occurs temporally when flickering heterochromatic stimuli fuse into an intermediate colour, even when there is no such pixel on the page. A more interesting phenomenon is a context gestalt, where the visual system integrates the context before attributing a particular colour value. Notice that in the image on the left, squares A and B seem to have quite different colour values, and even when they're united by isochromatic bars, that illusion persists because they do in fact have the same colour value. It is only when the context is completely eliminated by masking out all the background, such as I've done in the insert on the left of this page, that we can see that the colour values are in fact the same. All of these various forms of Gestalt perception of colour, whether they be spatial integration, temporal integration, or context integration, are all non-linear. I'm not going to go into all the phenomena listed on this page or the others that are not listed. They are simply there to illustrate the fact that non-linearity is pervasive in every dimension of this Gestalt process. To answer the question, how did we come by such a bespoke visual system, we have to go back into evolutionary time. The image at the top of this page represents a timeline from 4.53 billion years ago on the left up to the present moment, and you'll see that most of the timeline is occupied by blue, indicating the period during which ultraviolet and blue light adaptations were most useful for visual evolution. Only a small section on the right of that timeline is the period during which long wavelength discrimination drove visual evolution. So the blue section begins with archaea, which appear in the fossil record almost 3.5 billion years ago as single-cell organisms that used a photosystem based on chlorophyll to pump chlorine across the cell wall in order to survive in a hypersaline environment. It wasn't until 1.5 billion years ago that a second photosystem appeared based on all-trans retinal, and this allowed those early organisms to make better use of wavelengths around the 500 nanometer part of the spectrum. And then around 650 million years ago, 11 cis retinal appeared, and that enabled organisms to reconstitute their retinal even in the dark. Further along, we had ciliary opsin appearing, which enabled organisms to inhabit increasingly diverse ecological niches. But that entire period of evolutionary history was all about light harvesting and gathering light energy under different conditions and avoiding wavelengths below 300 nanometers, which would cause cell lysis. But since the energy in a photon of 600 nanometers is half that in 300 nanometers, all of these adaptations involved short wavelength, high energy light. It wasn't until we get beyond the early stage of multicellular organisms that we start to see evidence of rapid evolutionary behavior. So around 500 million years ago, an evolutionary guerrilla war broke out known as the Cambrian explosion, and that was largely driven by the ability of some species to detect the direction of light, the presence of looming shadow or flicker, and respond in certain ways that were adaptive or enable what was once prey to become predator. And evolutionary scientists such as Gordon Walls, quoted on this page, have remarked that the vertebrate eye appeared very rapidly during evolution. It has really surprised many observers how quickly it was. But what's not mentioned here is that once vertebrate organisms evolved, the tegmentum soon appeared, and it has persisted throughout evolutionary history as a highly conserved feature of the brain. The next part of our narrative involves changes in the opsin gene populations over time. In this diagram, you can see that when vertebrate land invasion occurred, there were quite a number of different opsin genes, but by the time we get to 150 million years ago, a small placental mammal, the size of a dormouse, was living a nocturnal existence to avoid becoming dinosaur lunch. And because of its nocturnal existence, its retina became rod-dominated, and it conserved only the OPN1 SW and LW genes. So at the time when the asteroid hit the Earth 66 million years ago and made all those hungry dinosaurs extinct, those placental animals managed to survive, and 55 million years ago, when the Earth suddenly began to warm in a period known as the Paleocene-Eocene thermal maximum. Forests appeared, the atmospheric oxygen increased, and suddenly the environment for these animals changed so much that they needed to make long wavelength discriminations under dusk or daylight conditions in order to find fruit and ripe leaves. And it was that demand which may have led to the evolution of trichromacy. We know that the paleological record has evidence of a skeleton found in southern Egypt, which could be the common relative of all current primates. Around about 40 to 45 million years ago, so-called New World primates separated from Old World primates, setting up a separate population in what is now Latin America. The Old World primates persisted and evolved under conditions of increasingly dense forest, living under forest canopies for many millions of years. And over that time, the wild population of the OPN1LW gene continued to drift towards having more and more variants containing the amino acid substitutions that favored discrimination around the green part of the spectrum, enabling these animals to select the ripest leaves. But that by itself wasn't enough to enable trichromacy. It was also necessary for any single primate embryo to be able to select one particular gene to transcribe out of all the ones at the terminal end of its X chromosome, so that it would express only one opsin in any one cone cell. We can see in the diagram on the bottom left of this slide that the locus control region, labeled LCR, is the region that controls which of the red and green cones is actually transcribed. And that whole tandem array is located at the very end of XQ28. We can also see on the bottom right that the primate neocortex is evolving rapidly shortly after trichromacy appeared. So the neocortex started to represent the world based on the high resolution input that was available to it from long wavelength detection. At the top part of the slide, we've amplified the timeline on a previous slide, showing the neocortex development beginning after 55 million years ago. And we see that the old world primate trichromacy appeared 30 million years ago, and then as open savanna grasslands appeared, apes evolved, requiring more upright posture, longer limbs, higher density cone arrays in the central retina, and also the ability to represent the world by recalling which fruits and which locations were most desirable at a previous time. There are several other adaptations that were required for this bottom-up version of trichromacy to appear. Some of those are listed in the slide, but what's listed at the bottom, which is cortical plasticity, also points to the fact that this top-down notion of colour idealism, or the ability to represent colours and represent objects in, as it were, a mental blackboard, to compare them with memories of previous perceptions and to imagine what alternative futures should look like. All of that required capacities which are distinctly non-ocular, and it's as if the ocular input is simply plugged into that ability to think and imagine, but it's in that second capacity that colour perception actually exists, not in the process of bottom-up contrast detection. In this slide, we can see that the locus control gene is followed by more than one long wavelength variant in most normal humans. The commonest genotype is illustrated in the bottom left, where there is one red followed by two green opsin genes. And in this diagram, an error in replication has occurred so that the green gene has been duplicated. And this is a common event. On the image on the bottom right, we see that when the process of meiosis occurs and the two separate chromatids align it is normal for genetic information to be swapped between homologous genes. However, when that occurs between an OPN1 LW on one chromatid and an OPN1 MW on another chromatid, we get hybridization. And these hybrid genes are constantly being produced at a rate slightly higher in humans than in other primate populations. As apes grew into humans, the axial length increased from the size it was in that ancient ancestor 150 million years ago who was dodging dinosaurs, up to the present moment where the axial length is so much longer that blue light will no longer form a small spot size. And this is one of the reasons why the valuable real estate in the central fovea had to be vacated by those slow responding blue cones with very poor spatial contrast and replaced with faster responding L and MW cones with much higher spatial contrast. This is a schematic of the density of the blue cones distributed around the blue cone-free foveola in the human retina. And although the size of the tritanopic area in the central fovea may vary, it is widely claimed to be between 1.1 to 1.5 degrees. The maximum density of the blue cone area corresponds to a gap of around about 2 degrees from the middle on one side to the middle on the opposite side of the fovea, which means that if we want to be sure to have a colour vision test that can detect tritan or blue-yellow contrast, we should design a target that has an average gap of around 2 degrees, but the gap needs to vary between about 1.5 to 2.5. The C-test is a test for tritan contrast that has been designed with those principles in mind. It's been designed to remove as many possible inputs to the parvocellular system as possible. So there is no luminance contrast, no red-green contrast, and since horizontal, vertical and parallel lines are preferentially detected by the parvocellular system, they have had to be removed from the design. So the C-test is not a Landolt-C, but rather an oblique gap or a wedge removed from a foreground circle. And the final strategy to discourage parvocellular perception is to ask observers to look at that foreground circle and move their eye constantly in a moving pattern around it and encounter the gap dynamically and then identify where the gap is. This is the 10 plates of the C-test shown in one slide, where you can see that the gap is in a different location in sequential slides and is increasingly harder to find due to decreasing contrast. Plates 9 and 10 are duplicated to allow a different image for left and right eyes. Congenital anomalous perception caused by anomalies in either OPN1 LW called protan defects or OPN1 MW called deutan defects are sometimes mapped on a CIE chromaticity diagram, one of which is in the bottom right of this slide. However, in this slide, I have not shown the separate confusion axes, merely a confusion axis labelled as Ishihara, to indicate that it is in a different axis to the tritan axis measured by the C-test or other similar tests. In the case of acquired anomalous perception, the defect is seldom simply a red-green confusion axis, and in the case of inflammatory optic neuropathies, it is almost always more significant in the blue-yellow than in the red-green confusion axis, even though the red-green axis is the one most commonly measured clinically. In cases where the acute stage is measured, subjects may have a combination of both red-green and blue-yellow confusion, resulting in an anarchic axis. But later in the disease, the parvocellular system is more likely to recover than the coniocellular, in which case the Ishihara axis measurement may appear relatively normal, but if the tritan axis were measured, it could still show a defect at that stage of the disease. The Ishihara plate test was designed by Shinobu Ishihara using hand-painted watercolors with the assistance of his adjutant in the Japanese military. It was designed at a time when Japan was about to enter a world war early in the 20th century, and the purpose of it was to categorically select normals from abnormals with regard to red-green colour perception. It was never designed as a test to detect severity of red-green confusion or to monitor disease. The original hand-painted designs were later updated into what we now call the Ishihara plate test in the early 1930s, using so-called international recognised numerals. There are 4 different designs amongst the plates. There are the disappearing digits, the transformation, hidden digit, and qualitative plates. And there are 2 most common editions, the 24 and 38 plate series. Only the transformation and disappearing digit plates are actually reliable, and they number plates 2 to 13 in the 24 plate series, giving a total selection of 12, or 2 to 17 in the 38 plate series, giving a total selection of 16. The other plates should never be used. The reason that hidden digit plates should not be used is that most often the test is used in young people, and the younger the observer, the higher the false positive rate for those plates. In some of those plates, the false positive rate varies from 20 to 40% in very young observers because they have above average ability to find the hidden digit on the plate, something that older observers simply can't do. As far as the qualitative plate is concerned, the accuracy at determining whether an observer is protan or deutan is only slightly better than 50%, so one may as well flip a coin. In general, when a person fails the Ishihara plate test using the reliable plates, a second test is required to sort out whether they are true positive from false positive, because there are quite a few false positives with this test, and also to sort out are they protan or deutan. So if a person has a normal Ishihara test result, no further testing may be required. But if a person has an abnormal Ishihara test result, a second test is almost always required. The design consists of one of 12 pigments mixed in various patterns, with a total of 6 or 7 per page on any of these plates. Because the total color palette is limited, the total dynamic range of color difference across the series is actually very small, and the majority of errors made by observers are in fact due to non-colorimetric differences. The cutoff score depends on the population being tested. If one is testing women with a prior probability of only 1 in 200 of a true positive, then finding up to 8 false positive results can be quite troublesome. Whereas if one is testing Caucasian males, where the prior probability is 6 to 8%, or Black African males, where the prior probability might be 2 or 3%, then a false positive rate of 8% at some age groups may not be a major problem if a second test is readily available. The illustration on this page is an ROC curve that serves to illustrate the principle of finding the correct cutoff score. The greater the number of plates, the more definite the cutoff judgment can be, which is why the 38 plate series is preferable to the 24. But with both series, the correct cutoff does depend on the prior probability, since there is a value associated with false negatives and a value associated with false positives, and the person performing the test needs to be aware of those values and adjust the cutoff accordingly. When applying Bayesian principles, it may be necessary to apply a cutoff of 5 or 6 for females, a cutoff of 4 for the 38 plate series in males, and 3 for the 24 plate series in males, and those are common values. So why are there so many false positives in the Ishihara test? One way of understanding this is to consider what an anomalous observer actually sees. That is to say, what is the transformed image that a person with red-green anomalous colour vision actually sees in the transformation plates. One way of predicting this is to perform masking tests on the numeral 8, where quite a legible 3 appears, or on a 6, where a less legible 5 appears, or on 29, where a highly illegible 70 might appear, or some other number. Also, if you consider the form of 1 that results by masking a 7, it's quite different to the form of 1 that results by masking a 4. People with an above average vision are often distracted by the large serifs and other complexities in the graphemes, the fact that the sizes of similar numerals differ between plates, and that there are distracting elements that are randomly arranged around critical points in some plates, and observers find these features more visible than the colorimetric differences under certain conditions. In fact, the colorimetric differences across the plates are remarkably small. And one way to see this is to take the full set of transformation and disappearing digit plates, which I've done in the top part of this graph, and then overlay the short wavelength filter. And you'll notice that all of the Ishihara plates in the top section of the image are roughly equally affected, and they're all still legible. When that is done to the C-test in the lower part of the image, it's only really the first couple of plates that remain legible, and the effect is not linear across the test series. If we do the same with a long wavelength filter, we get much the same result, although in this case the Ishihara plates are a little bit more legible. And what this test also shows us is that testing for Tritan contrast, one has to be far more careful about the illuminant and to note whether or not nuclear cataract exists than one does for testing with the Ishihara plates. So what to do with an Ishihara score? If we go into all the trouble of using correct illumination, using the greatest number of disappearing digit and transformation plates we can lay our hands on, and so we get a score out of 16, and we think we've done an excellent job. What does that score mean? Well, it is simply multiple similar categorical test results. It is not an ordinal or an interval score. It can't be ordinal because there is no hierarchy of difficulty through the set, and it's certainly not interval because we're not measuring in any significant way the amount of confusion. Now, a categorical test would be a test in which we didn't measure the age of a population, but simply recorded whether the members of a population were adults or children. If that's all we have by way of data, we certainly can't work out the average age. And yet, that is the type of process which is often done using the Ishihara results. The categorical result, reinforced by numerous repetitions to be highly probable, is taken to be a surrogate of an ordinal result, which is mathematically invalid. The Tritan contrast testing has a number of uses in professional life for ophthalmologists and also for neurologists. Tritan contrast testing is especially useful in amblyopia, retinal vascular disease, and optic neuropathies. In amblyopia, the presence of a pathway-specific inhibition in the presence of developmental retinal rivalry means that the bottom-up parvocellular signal is selectively suppressed at the lateral geniculate nucleus, so that coniocellular function is normal in people with amblyopia up to 660 or even worse Snellen acuity. And it's useful because this is a positive test for amblyopia, unlike the normal diagnostic criteria, which is simply a diagnosis of exclusion. Another condition in which Tritan testing is useful is retinal vascular disease, because the parvocellular system is especially affected by edema, but less sensitive to ischemia than the coniocellular system. The reverse is true for the coniocellular system because the spatial density is so low that edema hardly affects its performance. So anyone who has a very low Tritan contrast score has a poor prognosis after central vein occlusion or in diabetic retinopathy. Although optic neuropathies are not uniform as a group in the way they cause colour vision defects, most inflammatory neuropathies do involve blue-yellow contrast to a greater degree than red-green. One problem with generalizing about optic neuropathies is that glaucoma is a standout exception, where the use of the C-test, for example, is relatively uninformative. However, in most acute onset optic neuropathies, due to nutritional deficiency, toxins, or inflammation mainly, the blue-yellow contrast defect may be more profound than the red-green and is less likely to recover afterwards. So it's useful to have a Tritan score that is ordinal as a way of measuring a person's progress over time and knowing when they really have recovered, and also one that has a very low false positive rate. The Ishihara test is not ideally suited to this task, but is often used solely as a form of colour vision testing under those conditions, which seems slightly anomalous. So in conclusion, we've looked at two different metaphysical claims for what colour perception or colour consciousness really means. One is physicalism, the other idealism. And what I've tried to suggest in this talk is that it's a mistake to say that colour consciousness can be explained by physicalism, since consciousness is by definition a top-down process. However, it would be a mistake to say that idealism in any way contributes to a person's ability to separate similar clues on a test page or to match colours in a clinical colour vision test. That is definitely the result of bottom-up skills that an observer has and can be applied monocularly in the case of acquired diseases. Let me tell you a brief story. Imagine that we had a video of some ancestral primate who was in a forest foraging for berries and ripe leaves. We might watch them select a ripe fruit in the shape of a berry from a bush and say, well, that is an evolved capacity that having high quality red-green contrast has suited them to do, and we can see why that's an adaptive evolutionary trait. We might then see them climbing through the undergrowth, picking the ripest leaves from a densely matted foliage, and we might be quite impressed that nature has equipped them with the ability to send both high luminance contrast and high red-green contrast through the same parvocellular system as a multiplex signal, because in some ways that could provide useful short circuits for the processing that enables rapid responses to clues about chromatic difference in the region of 500 to 550 nanometers, where it is most important that one looks for long contrast difference under those conditions. However, if somewhere in the peripheral field of that primate there's a dark colour that seems to be moving through the undergrowth, and that dark colour is illuminated alternately by direct sunlight, by reflected light of plants, and at various times has the blue of a lake in the background or the green of the foliage, then if that primate's colour perception was purely driven by bottom-up adaptations, they would see a number of disconnected sequential stimuli rather than a line in the undergrowth. And it's only an ability to detect the line in the undergrowth that gave them the ability to pass on their genes. And so I would like to suggest that that unique adaptation is extremely useful for colour consciousness and for the logic of behaviour, even though it is not especially relevant to how a person in a clinical setting makes hue distinctions. So ophthalmologists are ideally placed to correctly apply and interpret colour vision tests, and I hope that this talk will help people to reconsider the use of colour vision testing in their clinical practice. For anyone who wants to know more about the C-test, this is a list of references and a download link where you can either download a PDF copy in a printable format or perform an online version of the test. Thank you for your attention.